Cool. Excellent. We can start. So, anyway, quiz was over. I'm expecting your results today. You will get, I expect you'll get 100%. Yeah, I will be very sad if you don't. It was so easy quiz, like, yeah. No, please get 100%. I'm kind of nervous. I'm probably more nervous than you are. Because I really wanted everyone to as you so you saw now in the first, so I'm not planning to mess up on next quiz, so it won't be hard. Quizzes are there for you to be fast knowledge check. So from now on, quizzes will not be 1 hour. I just gave you 1 hour now to you, so you can adapt on me and I adapt on you. So as you could see, and I saw, most of you finish in less than 30 minutes. That's the thing. And you again, I don't know the class. It's my first time teaching this class, and I'm trying to see what's. I don't want to give too much. So for next quiz will be more. I think it will for me will be harder. Like if I would do quiz 2, for me that would be the hardest because I really hate memorization. So there will be a lot, like there will be more organisms that you will have to memorize. But I will tell you patterns, how can you easily memorize, and I will help you to like narrow down the amount of organisms that you memorize, because I want you to bring something home when you finish this class, not something that you will forget. And everything what I'm going to talk about today will be, I will give you something interesting that you can memorize. So, and maybe will be a little more questions. Well, if I stay to 45 minutes, then I won't increase number of questions. So it will be like 24, 30, yes.
Well, maybe to get our quiz a little bit fast so that you can see where we went wrong.
Yeah, I can definitely. So I'm very fine with that. What I can do, so I avoid you guys chasing me around, I can maybe leave quizzes in front of my office or in front of my lab, and I instruct you where to come. Because I don't know where I will be. I work in the lab. I am out in. Yeah.
Could you bring them to a lecture?
Well, I could, but then in that case, it will take a lot of time from the class. I can, but maybe I leave it over there, and then you pick it up on. I don't want to take time from the class. So I can do that. I have actually there in my file, so I can bring it to the lecture. That will be easier for you. But what if somebody is not on the lecture? So I will do both. So I will bring it here and leave on this. There is one chair or desk in front of the classroom, so I will leave it over there, and then you guys collect it. But try to do that before class, so we don't take class time. And for those people who are not there, I will announce. I'll leave them in front of my lab upstairs on the 5th floor of microbiology wing, and then you can come at your convenience and take it back. So I don't think anyone did anything wrong. I'm curious. I don't know where you make mistakes. No. I don't want to think about mistakes. Any more questions? Yes, Ricky.
Do you have bonus questions?
Yes. I always give bonus questions. Yeah, I love bonus questions. They're very nice way to get extra points. So I always have that. That's my. If there is less people, I would give midterm take home. But I cannot read 200 midterm questions. No way. Never, ever. I would probably shoot myself. So no. I already have like 29 students from micro 311, 39 from 315. I already see myself, I'm recording. I already see myself getting my 6 buddies from near store and going home marking your midterms. And I cannot really use it, but yes, my 6 buddies, you all know what I mean. Yeah, so I can't do that with you. There are 200 of you. So that's why Nora said Scantron, only Scantron. Not for midterm, for midterm, God, I would kill myself. Yeah, okay. So probably midterm you won't get in 1 week or so. When is the midterm? Somebody, is it November 9th? that is before reading week. Okay, then all good. Reading week, the reading week it is. I know that this one, that okay, that's why I structured it like that. So I have you after the reading, I have you after the, like before, when reading week starts, so I can mark you and I get rid of those kids from 2 classes before. Yeah, I knew I did something very smart, just I couldn't remember what. So, well, not last, but you all heard about AOBs and MOBs. I posted that lecture. It's a very short lecture, and there are only a couple of things that I want you to memorize. That's about AOB, MOB, evolutionary connection. But now we will talk about, so honestly, this is my favorite lecture in this unit because we talk about other distinctive chemotrophic bacteria, and I will talk about bioluminescent bacteria and predatory bacteria, which are amazing organisms, then asymmetric cell division. So this is very easy to memorize. And that's why I love it. I don't really like memorizing a lot. I like to understand. So today we will, so what means chemotrophic? So chemotrophic means using special chemicals to grow. So that means chemotrophic. And those special, so iron reducers are also chemotrophic organisms. So they can be chemolithotrophic, but that's basically they can be chemooligotropic. That means they use they need very small amount of energy to grow, but they are still chemotrophs. So they're all of these listed here, which are iron reducers, iron oxidizers, manganese reducers, manganese oxidizers. So they're all chemotrophic organisms. They all use special chemicals, which are metals in this case. These bacteria, predators, stalked and bioluminescent, they're not chemotrophic bacteria. So they're special groups of bacteria. I'm talking about them in functional diversity just because they are usually covered in this type of classes as a part of morphological diversity. So because morphological diversity is a special sort of diversity. Now, I personally think that morphological diversity is part of functional diversity because functional diversity is something that is related to the form and function. So based on your form, you will have different function. So that's why I didn't want to have separate. Initially, I had separate units like morphological diversity, but then when I went back through that after I finished lectures, I was thinking that this actually doesn't fit in a separate unit, so I would cover it as a special group of bacteria because they're so nice that I couldn't really not cover them. They're very interesting. Now, iron reducers are the first group of organisms that I will cover, and we heard about them when we talked about metal, like when we talked about different types of metabolism, and what they do is literally they are coupling reduction of oxidized metal to the cell growth, which means they have to have this iron in ferric form 3+. So that's oxidized form. Reduced form is the one that has more electrons, which means it's less positive. It's iron 2+. So they respire iron 3+. So they oxidize something else. They use electrons from something else, and they deliver those electrons to iron 3+. So iron is here electron accepted. The reason why I'm pointing this out so much is because the iron can be also electron donor. So it's important to be in 3+ form to be electron accepted. And these organisms are nice because they're very unique in the ability to respire solid metals. Who can do that? Well, they can do that. Here is how. So this basically reduction of this metal, like respiration of metal, occurs during the cell growth. So is that so? One might ask, is that the same reduction of metal that happens in cellular metabolism during other processes? Because you will have oxidoreduction processes in all organisms. This is not the same, and it's not the same because the difference is when they respire metal, they generate energy. When you have other type of metal reductions for other purposes, there is no energy generation. So that's the difference. So it's not the same. It's just respiration of metal that results in energy production. Now, these iron reducers are very phylogenetically diverse. And starting from unit 4, which will be week after, not next week, we will talk about proteobacteria, like diversity of proteobacteria, and then we will talk about, oh, yes? Oh, sorry. Bless you. No worries, bless you. Whoever sneezed, bless you. Sorry. We will talk about all these bacterial phyla, proteobacteria, Firmicutes, Bacteroides, Actinobacteria. Proteobacteria are the most diverse phylum. So literally in that phylum, you will see all types of metabolism. Everything is there. So others, slightly different. That's why probably proteobacteria is the hardest one. So iron reducers, you will find them in proteobacteria. So phylogenetically, they are proteobacteria, but they can be in other phyla as well. The ferribacter is probably the most interesting one. You will recognize it based on the word. The ferri. This stimulates iron. Ferro stands for iron. Firmicutes acidobacteria. So those are 4 phyla where you will find iron reducers. And of course, in archaea will be in Crenarchaeota. Archaea are easier. There are like 6 phyla that you have to memorize. And Crenarchaeota are usually everything is in Crenarchaeota or Euryarchaeota. Now, interesting about iron respiration is probably it existed in last universal common ancestor, Luca. And that makes sense, because if you think about what was present in early Earth, a lot of iron, so whatever developed had to be able to somehow make a use of that available iron. So yeah, this makes sense. Unlike nitrogen fixation really doesn't make a lot of sense. But again, I'm not going to dispute on that. That, as you heard in lecture, I do think that nitrogen fixation was after denitrification, but not many people think, not everyone thinks like that. So probably iron respiration was one of the first evolving processes. Again, just if you think about how early Earth looked. And then later on, iron as lineages is separated from Luca, and then each of them developed independently. Iron respiration was lost in some organisms, again, based on in which environment they evolved. So gene probably got lost. What are characteristics of these organisms? So they use insoluble external electron acceptors. So iron oxide, that is insoluble. So what they have is what they have to have is they literally have those outer membrane extensions, which I mentioned in Geobacter and Shewanella. And Geobacter and Shewanella are among iron reducers. So those they have the cytochromes are proteins that participate in respiration, and these organisms, iron reducers, they have outer membrane cytochromes. They're like outer membrane extensions. So those cytochromes are literally piling up and making something called nanowire, and then that nanowire makes a contact with electron acceptor, and that's how respiration of metal happens. Most of them are anaerobics, and which means they're facultative. They are not facultative anaerobics. They are obligatory anaerobics. That means they cannot use oxygen to breathe. They would die. They can use iron and other alternative acceptors. In terms of electron donors, so iron is acceptor, so like insoluble iron. Donors can be something organic. Like organic compounds can be various, fatty acid, alcohol, sugar. That's all organic, something that can extract energy. And if you think about tower of power, iron would be here, organic compound is here in terms of energy. So they have to take something that can be higher energetically. And there is no carbon fixation. This is their characteristic. Autotrophy is not present in these organisms. They're completely organic carbon dependent. They cannot fix carbon dioxide. Some of famous iron reducers, again, Geobacter is, you also had that question in exam. Geobacter is the most famous one that I will show later. It's used in commercial purposes. And the other one is Pelobacter. The other two that are hyperthermophilic are Thermos and Thermatoga. They are adapted to high temperatures, but Geobacter and Pelobacter are growing optimally at ambient temperature. Geobacter is the one that contains pili, like external structures that facilitate energy transport to the electron transport to the solid acceptors. And those pili actually contain those cytochromes that interact like nanowires. So in pili, you will have those cytochromes. Looks like a little extension or through cell membrane that makes a contact with outer, like an insoluble iron oxide. Pelobacter is fermentor, so that means it uses lactate as electron donor, and it can use also elemental sulfur as acceptor in addition to iron. So again, alternative acceptor is possible, but cannot be oxygen, can be iron or sulfur. The next, so we will stay with reducers, like manganese reducers. So later on you will see that I moved, so I'm talking about iron reducers, and then I talk about manganese reducers, and then I will refer to metal reducers. So as you probably noticed, bacteria or microorganisms will respire whatever is there. So some of them can use, there is an organism that I will mention later that was identified as an iron reducer, but then they did analysis and they noticed that it becomes manganese reducer when you overwhelm it with manganese. So they just have ability. That means the gene for manganese reduction was not lost during evolution, just requires a trigger that will cause that gene expression. And that's what actually fascinates me. That's fascinating about bacteria, about microorganisms. They will get energy from anything you give them. Some of them you can kill, which is great. So in terms of manganese reducers, what does that mean? That means that they're coupling, they have to have oxidized metal like manganese, and that reduction of manganese will cause formation of proton motive force, like energy. So metal here is energy acceptor, is electron acceptor. So the same like iron reducers, they evolve the ability to respire solid metals. That's pretty much unique in, I don't think I ever heard for any other organism to does that. So bacterial manganese reducers are very diverse, and there is also only one archaeal phylum, but while iron respires are mostly in Cranarchaeota, in terms of archaea, these are Euryarchaeota, so different archaeal phylum. It will be much more clear when we talk about archaeal diversity. So again, iron reducers are present in Firmicutes, in proteobacteria, and you will see also here Firmicutes and proteobacteria for manganese reducers. Actinobacteria are famous manganese reducers, acidobacteria, but you have acidobacteria in iron reducers. And in iron reducers, you will have in iron reducers, you will have that Deferibacters, which is pretty common for iron. Here you have Bacteroides. So I'm just trying to teach you how to memorize all these phyla that have different reducers, because for next quiz this will come into the question on questions. So just find some logic. Again, proteobacteria is always there. So that's pretty much that's the most diverse, like mitochondria in eukaryotes. Everything is there. For archaea, it's a little maybe harder because you have to actually know. I have only one example of manganese reducers. Actually, no, I have one. So there, I had. So again, ANME. What is ANME? Anaerobic methanoxidation. So I had very nice question from a student who said that ANME is kind of mystery because they didn't understand is ANME methanotroph? Is it methanogen? And the thing is this, it was initially discovered as methanogen. So it's archaea, makes methane. But later on, scientists discovered that it actually can also do something called reverse methanogenesis, which is methanotrophy, which means oxidize methane as a food, but respire something other than oxygen. And it can respire iron, you will see later. But recently they discovered that famous manganese reducer is actually ANME. So it doesn't work with SRBs, like those ANME that we covered in first section. It works with, it just grows on its own when you give them manganese. And this is actually from the recent study that I can send if you want. These guys discovered this family Methanopteridans. So Methanopteridans, which is called manganese reducens. So initially, this family is known to respire iron or nitrate. But then what they found, so the way what they did is basically they took inoculum from fresh water, in which they knew it contains ANME, the one that respires nitrate and iron, and they took this inoculum and put it in the fresh inoculum into the bioreactor. So literally environmental sample into the bioreactor, and then they added manganese, and the form of manganese is called birnacid. That's manganese. Manganese oxide. So they added that form of manganese into the bioreactor, and then they took a sample at the beginning, which is this first one at 0 hours, and they did a metagenomic analysis of sample. Then they kept taking sample at defined time point, like 60 days, 2 months. So basically, it was actually a very long experiment. And you can see how population is. I don't know how much you can see here, but what you can what they saw first that it was dominated by iron, by this ferrireducens. So methanopterans ferrireducens, which is a purple. You see at the beginning, and like this the big purple line. And as they kept taking sample, they were still adding manganese. So basically, they saw that population community in that bioreactor actually changes from the one that was ferrireducens. It was actually manganese reducers. So when they did sequencing, they saw that ferrireducens and manganese reducers actually have they did 16S RNA sequencing, and they analyzed it. They saw it's not the same species. It's a different organism. They, of course, are belonging to the same phyla. They are both ANMEs, but they use different terminal electron acceptors. One uses iron, the other uses manganese, and population is definitely changed. Now, basically, manganese and iron are a very nice source of energy for organisms. And as always, we as people, we exploit all characteristics that organisms have, and we employ this into the microbial fuel cell. So that's what I will cover in next few slides. So the way how they do, generally the way how they do this metal reduction, and this is what we use in microbial fuel cell, is electron shuttle or direct delivery. So they have to have those outer membrane cytochromes, and they have to have a mode to deliver electrons to that oxide, the solid one. So it can be through shuttle, and shuttle is humic acid, or it can be direct. That just means no humic acid. So the way how that's done is basically they oxidize some organic compound in cytosol. Can be acetate. I gave acetate as example. Doesn't have to be acetate, but it has to be organic. Then electrons are extracted and sent to membranes, and there are those membrane extensions that are called nanowires, like cytochromes. So cytochromes make nanowires, and some organisms have this humic acid, which is literally soluble electron shuttle. It is like a shuttle that takes electrons and drives them to the external electron acceptor, and that's one. when there is no some organisms just don't use humic acid. They directly deliver electrons to the membrane. And this is what we, as people, exploited in microbial fuel cell. How many of you heard about microbial fuel cell? Didn't? Oh, good. People in 311, they all know about this. So that's this is the way to generate electricity from microbes. And the way how it functions is basically like you have two compartments in microbial. So microbial fuel cell setup has two compartments. One is anoxic with anode, and second one is oxic with cathode. So what you will do, anode is usually made of iron oxides. So that's where you have your iron or manganese, but usually iron, and that is terminal electron acceptor. So that's where microbes will respire. So you will, what you will do, and in cathode, it's oxic. So in cathode, you have like regular oxygen. So what you will do, you will inoculate your iron reducer, like geobacter, into an oxic compartment, and you will give them some organic compound, whatever, like acetate. So they will extract electrons from acetate, and they send those electrons to the iron, because to the anode, that's where iron 3 plus is, and then it reduces iron 3 plus to 2 plus. Then those electrons are now driven through anode to cathode. Cathode is in oxic area. And in oxic compartment, those electrons end up at oxygen, but creating water. And that remaining energy is actually used to turn on light bulbs. So basically the energy is captured in oxic compartment. So that's how we actually exploit metal reducers. Famous metal reducer, geobacter, which this time I will ask way more on the exam, on the quiz, because we are getting more into the harder stuff, is organism that has those specific membrane extensions made of cytochrome OMC. It's enough to know as OMC proteins. So they make those electroconductive pili. They're called electroconductive because they get electrons and move them through that wire. That's why we call it nanowire. So this is how bacteria looks. So you can see these electroconductive pili that make a contact, that can make contact with the iron, and they also have these additional extensions, which again help them to establish contact with iron. Some of the famous organisms that are used in microbial fuel cell are, and you will know by name, ferrireducens, Rhodopterax. It's enough to know R ferrireducens, because when something is ferrireducens, that means it reduces iron. I would personally like if you know Pseudomonas aeruginosa and Clostridium butyricum. Those are famous, and that would be at least 3 organisms would be enough. And Pseudomonas aeruginosa and Clostridium butyricum are famous organisms anyway. So with Rhodopterax ferrireducens, this will be 3 most important organisms to know. Another interesting note that I recently found is Synecocystis, which is cyanobacteria that is employed in microbial fuel cell. I had no clue that you can have cyanobacteria. I always thought it's only iron reducers. And the reason why it's discovered is because it also produces nanowires. So, and it's discovered that it can generate electricity. So what Synecocystis does, it's actually consuming carbon dioxide as a consequence of glucose oxidation. So one thing that I didn't really understand here is, and I will leave that open, I still didn't, that paper doesn't talk about that. So Synecocystis is cyanobacteria that I will ask about. In one of next classes, I will talk more about Synecocystis. It's important cyanobacteria. When we talk about proteobacteria, we will talk about that organism. So this organism cannot live in an oxygen compartment because it will produce oxygen. So probably, like, where is this organism growing in? Is it in oxygen compartment or anoxic? They don't say it. I think in oxygen, but I'm not sure. The paper doesn't say. To me, it makes sense that you would put this organism into where cathode is, but I'm not really sure. Any questions before we move to the iron oxidizers? Yes.
Yeah, I'm just wondering, why does it produce the nanowires if it's not like reaching up for an oxidator?
Good question. I also don't know that. That's what I tried to search. Why would they make nanowires if they don't have to respire metal? But they don't. They are phototropic organisms. They produce oxygen. They consume carbon dioxide. So they don't respire iron. It is very good question, and it's something that I was also wondering when I found this paper. It's like, why would they have nanowires that are developed for the purpose of respiration of solid metals if that's not what they do?
So nanowires are transport electrons to the next structure.
Yes.
Is there anything like into the structure for like depositing or something?
You know what could be? I don't think it's waste disposing. Maybe, I don't know, but I think it could be for maybe motility, like making a pili that helps you to move. So it's possible because Synecocystis is phototrophic organism that evolved to capture light. So maybe it made nanowires like a pili, so it can migrate where light intensity is higher, so it can capture better light for photosynthesis. So that might be the reason why it has it.
Oh,
Ask, please, I love it.
Yeah, I would feel like, why would you call it, what was the characteristic in the paper that made them call it a nanowire if it's not specifically transported?
Because it's made of cytochromes, and usually pili are made of different proteins.
Okay.
So if you see pili made of cytochromes are usually involved in respiration. So you would think that these cytochromes must do something with respiration. Yeah. Thank you for question and please ask these kind of questions. I really like when you ask questions. And yes.
Is it possible that in that case that their faculty
Yes. So what's your name? Sorry. Diana said, is it possible that they are facultative iron reducers, which means that if there is nothing else, they will respire iron? Yes. Why not? It's very possible. They apparently have gene that helps them to produce something that will help them to respire iron. So yes, it's possible they are facultative iron reducers. Yes, Jay.
Just to check my understanding, so the iron reducers can respire manganese if they're overwhelmed, and the manganese can respire iron if they need to?
So that is okay. So I cannot say that that's quite correct. What I can say is that those people saw change in community from iron reducers to the manganese, and when they did close sequencing of 16S, they found differences between iron reducers and manganese reducers. So I don't think that their point was community changing, but I personally think that iron reducers would be able to adapt to the manganese and to respire it if it's overwhelmed with it, because they have all structures required, and there is energy difference. Like organic compound, manganese and iron are very close in terms of energy, so organic compound is there. So yes, I think they could. But I don't have scientific data that I know.
Okay,
please.
If they were facultative, what conditions were they growing them into that would cause them to start, exhibiting like the iron respiration gene? Like, why would they produce them?
I could think of maybe if they evolved in early stage, which they did, same like Hostillaria limnetica that what that got exposed to the compounded block 1 of photosystems. So it had that organism had to learn how to extract electrons from sulfur instead of water because it cannot use other photosystem. So possibly that they got exposed to the excess of iron and without anything else, without light, with excess of iron, maybe they directed cytochrome production toward the membrane to send electrons toward the external acceptor rather than to photosystems, because maybe photosystem was not expressed when there is no enough light. So that would be, again, this is just my, we call this educated guess. I'm just thinking logically. I don't have any data that I can confirm, but Emily, right? And God, and. I'm so bad at names. Please correct me. I hope I can memorize all of you, but I'm not so smart. So yeah, that's my educated guess. That like there was no more light, so okay, let's direct all quinone, cytochrome, everything toward the membrane, make extension, and get to anything that can respire metal. And then again, as we know, this feature is favored by selection then, because it helped this organism to survive in the conditions where nothing else could survive, and then it just got transferred to next generation. Again, my educated guess.
And the paper didn't include, like, it wasn't specifying the condition for it.
They just talk about I can send you, I actually paused the paper on class. It talks about conditions. So what the point of the paper is how they made electricity in microbial fuel cell using cysts.
Oh.
So that's the topic of the paper. So when you write paper, you actually, maybe they got some results that they didn't include in paper because it didn't fit in the story. Like that's very often in science. Like you generate a bunch of data, but they don't really tell you anything that you can share with your reviewers, so you just keep that for yourself and your students, and you share only something that tells the story, that is correct, but tells the story. So there is no, like I had tons of data that nobody will ever see because it just didn't fit into my story for my papers. So, but I will put that paper on e-class, please, and remind me if I forget. Questions? All easy, huh? Yep. Oxidizers. So again, iron can be you can have reducers and oxidizers. So iron can be in both forms, but if it's oxidizer, then there must be some electrons to extract, like 2+. That's why it's oxidizer. Now, in this case, we talk about iron as an electron donor, and as everything iron related, this probably evolved very early in Earth history. And this, so it's still iron, but it's a different form of iron, and different organisms use it. So it won't be iron, it won't be ferry reducer anymore. And these guys are strongly affected by pH and oxygen, and there is a chemical reason why. Because at pH 7.7, which is like slightly, well, neutral, basic state, neutral. So at pH 7.7 with oxygen, iron gets precipitated, and it's not available for microbes anymore. Stable iron is only on pH 6.4, and iron could be stable only without oxygen present that was in early Earth. So later on, it was really reactive, and it couldn't be really used by microbes. And unlike iron reducers, iron oxidizers are way more complicated, and there are 4 different groups that you have to know. It's easy to memorize them because they are either anaerobic or aerobic. So, and then there are certain modifications. So if they are aerobic, that means they are acidophilic. That means they love low pH. If they can also be aerobic, but thrive in neutral pH, like 7. They're easy. Neutrophilic iron oxidizers. If they are anaerobic, they can be phototrophs, or they can be chemotrophic. Phototrophs are easy. So we'll go step by step. So aerobic acidophiles they're iron oxidizers that you will always find them somewhere close to the as like a mine drain where you had mines like acid mine drainage. That's where you will find them. It's like a very low pH, and they can use elemental sulfur to survive. So they will oxidize iron, take atoms from iron, but they respire sulfur, elemental sulfur. So they will be like sulfur reducers, and we mentioned them in a previous units. So ferroplasma is the famous archaea that I really think it's not hard to memorize, and leptospirillum and acidotheobacillus is easy to memorize because it's acidotheobacillus. So what does that mean? Acido, acid. Thio, that's for sulfur. It respires sulfur, and bacillus is bacteria. So bacillus bacteria. Now, these were probably very old because, as you see, they use iron sulfur, very present on early Earth, very present in anoxicum. That's why they are probably very, very old. Ferroplasma is my favorite organism that are here, and it's Acidotheobacillus is probably my favorite bacterium. Neutrophilic organisms have specific, they're different than those that live on low pH. At neutral pH, iron gets solid. So they have to have specific metabolism. So this organism is important to memorize, Gallionella. That one has this stalk structure that is made of iron. So it literally forms this twist stalk structure made of iron. And when iron is oxidating, like iron 2 plus, it creates iron 3 plus, which forms this stalk. So as it respires iron, it makes this stalk. And then iron hydroxide is excreted. This organism is autotrophic. It can fix carbon dioxide via Calvin cycle, but this twisted stalk is something that it's a very specific structure that I never saw that something similar in other organisms. So Gallionella literally accumulates iron and makes stalk that is a part of its structure. So anaerobic chemotrophic iron oxidase. Yes.
Sorry, you said iron is solid.
Yes.
But then earlier you said that iron precipitates
So precipitates at 7.7. I think it would be more correct if I say that iron precipitates at certain pH. Because what is like, is 7.7 neutral or base? I would, from my perspective, I think it's neutral. But for iron, it gets precipitated. So it really, I think that's why pH matters so much like for my organisms, 7.7 is neutral, like they will grow nice. But for some, like there are organisms that like to live at 6.4, which is still neutral, but 7.7 will kill them. It will be too big for them. But does that answer your question? Okay. Yes.
You mentioned that the stalk is formed into the structure, but then it also says that
it's excreted.
Yeah.
Okay. Yes. I agree. That's something that I am not. I'm pretty sure that this can be seen on microscope. So when what they do, they take iron inside of the organism to respire, and then that iron. Let's go to that slide. So literally, they take iron, they respire. They have to import it in the cell, so they respire it. So they don't do this like extracellular. They don't have nanovirus. So iron is inside the cell. So iron is 2 plus. Then it oxidizes. It takes all electrons, form 3 plus. Then my understanding is that that's how I got it from the book. I didn't see it on microscope. As they're forming it, so they're literally forming that iron hydroxide that grows like inside of the cell. So it's like a twisting that makes it, and that's that happens while it grows. Probably when cell divides, like in cell, that's again my educated guess. When cell divides, that hydroxide somehow gets exported into the external environment, because then cell has to make a space to take new set of iron and make new stuff to as new process of division start. So I agree that is kind of contradictory, but I think if we could see this under the microscope, if we could record it in a real time, it would make more sense. I didn't see it. I just read the book, and this is my understanding. That's how it functions. Basically, like it forms talc, like it respires, it grows, forms talc, cell divides, hydroxide is there, it gets exported, you import a new set, and then you start new. That's my understanding. I might be wrong, but it is something that we can look into. And thanks for the question. I agree. It is a little bit contradictory. I don't know, does that answer your question? But at least gives you something to search. Okay, more questions. Thank you for the questions. Finally, you guys start asking me. So I really like that. If I don't see someone's hand, you just yell. Okay. So now we move to the anaerobic iron oxidizers. So they can be chemotropic or phototropic. So if they're chemotropic, they just respire nitrate, NO3 minus. God, I apparently don't know how to pronounce this, nitrate. Famous genera are Thermomonas and Aquabacterium and Acidovorax. There are only 3 of them that you need to know. And more important is to know that they're in Proteobacteria. So that's way more. And it won't be very hard to memorize. Like Aquabacterium is pretty much easy to memorize, water bacterium. And these are, so why are these so interesting? So this is the paper that I found this summer while I was making these lectures, called Nitrate-Dependent Iron Oxidation Potential Metabolism on Mars. They say it's very new. It's like paper from 2018, so for 5 years. So basically what they say is that this might maybe this metabolism exists in other planets, and they talk about this microbial nitrate-dependent iron oxidation, or NDFO. So they probably search for these bacteria, Aquabacterium acidovorax, when they search microbial community on Mars. I tried to get a guy who is working on NASA projects to give us guest lecture. He can't do it this year, so I was very sad. He had a bunch of stories to share with us. I have another guy who works on cyanobacteria who will give you a guest lecture on October 2nd, on November 2nd, sorry. But I really wanted these people who are working with microbes in space, because it's for me, that's a very interesting topic. I think they don't share many things with us when they explore Mars and other planets. Anaerobic iron oxidizers that are doing phototrophy, easy peasy. Rhodobacter peroxidans. Not like Rhodobacter, you remember we talked about Rhodobacter. It's purple non-sulfur bacterium. When we talked about losing a trait that was beneficial in the first lecture, Rhodobacter, that's the one. And Chlorobium peroxidans. So the name peroxidans is all what you need to memorize. It's Chlorobium and Rhodobacter. And again, fun fact, oops, sorry. Iron oxidizers are very interesting because they are called magnetotactic bacteria. They have magnetosomes. Again, something that looks like organelle, but it's not organelle. So basically what those iron oxidizers have is a large set of genes that code enzymes involved in iron oxidation, and they create magnetosomes. So magnetosomes are intracellular structure that exist in bacteria, and they contain lipid bilayer, so they remind on organelle, so same like anamoxosomes. So again, you see connections with eukaryotic cell. So people say they're true organelles. I will never call this structure organelle, because it's in bacteria. But they have lipid structure, they have transporters. So technically they resemble organelle, but I just don't think that you can call something organelle if it exists in prokaryotic cell, because organelle is way more complex structure than magnetosome. And the function of these magnetosomes is to sense oxygen. Why? So magnetotactic bacteria, or so they need oxygen to live. So like those organisms that have that are iron oxidizers, and they can use oxygen to live. So they oxidize iron, but they respire oxygen. So they are apparently in an aerobic area. But too much oxygen is very toxic for them. So they have to find a balance. So what they do, thanks to those magnetosomes, they orientate themselves toward Earth magnetic pool. And they do that thanks to the iron oxides in magnetosomes. So basically, those iron oxides are helping in magnetosomes, are attracting cells, thanks to those magnetosome cells are attracting each other. They are aligning into the formation, and they migrate together towards specific oxygen gradient, which is called magnetoaerotaxis. And these are, so this is just microscope, basically this is just how it looks under microscope. So specialized organelles for magnetic navigation into this is basically the chain of magnetosome in bacterial cells. So this is how it looks, like a black structure full of iron oxides. That's how, and they align together and then they migrate together. So why would cells do that? So basically, if cells don't have magnetosomes, non-magnetosomic, and when they migrate towards specific oxygen gradient, randomly moving in 3D space, but when they have magnetosomes, they align together, and it's like a more organized move, and they move in one dimension. So again, in Martian meteorites, like Alan Hill's meteoric, had particles that were found that looked like magnetosomes. So studies did show that this is magnetosomic chain, but the question was whether they were formed biotically or abiotically. So yes, there were magnetosomes, there was iron oxide, but the question there, if there is life on Mars, are those maybe magnetotactic bacteria? So maybe that's the way of life formation in that planet. Maybe the same how we had iron sulfur, maybe they had maybe they had iron oxide. So we don't know, but it's very possible. So this is what was found in meteorite. Any questions before we move to the manganese oxidizers? Okay, so now for manganese oxidizers, I don't think I have many things to say. It is an interesting metabolism. I will finally have a chance to move to the my favorite predatory bacteria. So what is so famous about manganese oxidizers? So basically this is a reaction between manganese and oxygen. So manganese is an electron donor, but they respire oxygen. So manganese oxidized, electron donor, oxygen reduced, acceptor. So what's the purpose of manganese oxidation? Because the amount of energy that they get between manganese and oxygen, if you look Tower of Power, it's not very high. So the purpose of, so then the question would be, why would microorganisms do that? So the reason why they do that is to make their own terminal electron acceptor. So to make something that they can respire later. And here is how they do that. It's quite interesting metabolism. So they have an enzyme, those manganese oxidizers have an enzyme that is called multi-copper oxidase. So that's a special enzyme that exists in these organisms. And what this enzyme does, it takes electrons from manganese, reacts with oxygen, then makes mangan oxide. And that mangan oxide serves as a mangan oxide serves as a protective coat. So basically, a reaction between manganese and oxygen forms this manganese oxide this black coat. And the famous organism that does that is sporulating bacillus. So how bacillus does this? So when bacillus sporulates, spore is basically a dormant state of the cell. So when cells are exposed to some sort of stress, some of them, bacillus is one of the famous sporulation guys, it makes spore. Then inside of spore, there is that copper oxidase that performs manganese oxidation, like Mn plus oxygen. And it's when bacillus is about to sporulate, when it's under the stress, manganese oxidation is very active. So it literally makes those manganese oxides inside of spore. And those manganese oxides now will form that black protective coat. And this happens because then what cells do, with that protective coat, they literally sink toward the bottom of the ocean, like that, because it's heavy. Now, at the bottom of the ocean, there is abundance of electron donors. Remember, that's where the life probably started. So bacillus now already made its own terminal electron acceptor, which is manganese oxide. And now spore can germinate, they can extract electrons from whatever is available on the bottom of the ocean and respire manganese. Yes?
Sorry, so manganese oxidation is a stress response?
So that's a good. So I would say I think it is, because if we talk about sporulation, so in this example, yes, because bacillus will sporulate when it's under some nutrient limitation, under some stress, and it's noted that in bacillus that is actively sporulating, and sporulation happens when there is not enough nutrients, they will have that manganese oxidation, and they form that. But again, the example is. That's probably happening. The example is that what happens in the ocean. That's the reason why they develop this manganese oxidation. So it can be said that it would be a stress response in this example, but there are organisms that do it for other reasons, which I'm not mentioning here. So basically, no oxygen available, you make your own terminal electron acceptor. So the reason for creating those insoluble manganese oxide is that evolutionary adaptation to the exposure to an anoxic zone where there is very low amount of terminal electron acceptors. So you make your own. So when you sink down, when there is nothing to respire, you already made one for yourself. So any questions? No? So I don't think I will have time to go through summary that I have in the that I made this. I always, you know, that I always want to let you go a little bit earlier so you can digest what we learned. So we will finish with these 3 types of bacteria, and then we will have summary in next class. Or if we don't have time, then I will just record it and post. So my favorite are these predatory bacteria. They have very, very specific form and function, and they're very functionally diverse. So, and you will see by names, no way that you will ever forget them. So you will, of course, proteobacteria. Of course, they are in proteobacteria. Everything is in proteobacteria and Bacteroides. But what I find very cool is that one of them is actually named Vampirococcus. So for me, that was very interesting organism. Like, who can forget that? It's a vampire bacteria. Like, no way that anyone can forget it. And then the last one that I like and that I used to work with is Myxococcus. That one is awesome. I named one talk that I gave a long time ago as bacteria that create, or bacteria with a social media profiling. It's like, they actually have a very active social life. So Myxobacteria, I love those organisms. Lizobacter, Dactobacter, and Delovibrio are also predatory bacteria, but Vampirococcus and Myxococcus are definitely one that I will focus on mostly. And of course, they are protobacteria, like everything is protobacteria. Vampirococcus and Myxavibrio. So I know you will correct me, you will say it said Mycavibrio, but actually in Latin it said Myxa, because it's in front of the A, and whenever C is in front of the A, I, and E, it's read as C. Again, unfortunately, I spoke Latin, so I learned it, unfortunately. But yeah, so Mytsa vibrio and Vampirococcus. So they are epibiotic predators. What that means when something is epibiotic? That means they have to attach to the surface of their prey. So they attack, literally they attack like a vampire. So when they attach to the prey, they literally suck out nutrients from prey's cytoplasm and periplasm. Dactobacter is intracellular parasite. It's not parasite, it's a predator. So it invades host and consumes cytoplasmic compounds. So again, depending on what types of, it can be periplasmic, it can be cytoplasmic, it can be surface. So epibiotic means it's on the surface. So this Dactobacter goes inside. Bdello vibrio, on the other hand, goes into the periplasm. So it doesn't really go quite inside the cell like Dactobacter. Lysobacter and Myxococcus, I love Myxococcus. That one is, they're known as a social predators. Very social. They can be very nice to you when they hunt you. So they use something called swarming behavior, and they swarm, glide, and they get the prey. And then the reason why Lysobacter is called Lysobacter is because it actually lyses its prey. So it gives to the prey something that will kill it. And Myxococcus is one of very interesting organisms that I learned about when I did my third master in Wyoming. Those organisms have special molecules that help them to distinguish friends from foes. So they're called the receptors TRA. You don't have to know this, just my fun story. So what they do, they use that TRA as a signal recognition. So when they are in the soil, which they usually live, they will, based on that, they will recognize the one that has same TRA and they won't kill it. If the organism doesn't have same TRA, then they secrete some compound that they deliver to them and kill them. What can happen is that some organisms evolved immunity protein. So what can happen is they just develop immunity protein when myxobacteria goes and finds them. They don't have TRA, good, I will give them toxin, but then the organism has immunity protein that degrades toxin and then it survives. So it's a kind of competition that exists in soil. That's why I find that very, very, very interesting. It's not hard to memorize, like no way that you will ever forget that. Myxobacteria form this very interesting, this is how it looks, this is actually the microscope. So it's a fruiting body, that's a structure of bacteria. So it looks like a multicellular structure, and technically it is multicellular, but they are each separate cell. So they just live together, which is kind of cool for me. It reminds me on those volvox, is that how it's called, that animal that floats in the ocean? So that's what it reminds me of. That's like a lot of cells. It doesn't look the same, but it's the same principle. So they literally glide across the surface and they secrete enzymes that will help them, extracellular enzymes, to obtain nutrients, or they can lyse bacteria if it's Lysobacter. These myxobacteria are known about that complex behavior. So usually they migrate together, and fruiting body contains myxospores. This is that's how they survive. They're special cells that are very resistant to drying. So not only bacillus can form spores, this one can too. Now, stalked bacteria I have only one organism that I want to talk here about, even though it's a complex structure. So it produces those cytoplasmic extrusions, so that can be appendages, stalks, or hyphae, but all those extracellular compounds are collectively called prosteca. Usually those extrusions are smaller than cell diameter, and it contains cytoplasm and cell wall. So those stalk bacteria usually contain those prosteca. They help them to attach to the certain extracellular matter. They are aquatic organisms, and those prosteca can also help them to increase the surface area to the cell volume, and it actually helps them, protect them from sinking into the anoxic zone because they are oxic organisms. There are two organisms. One of them we mentioned already, Gallionella. So I talked about iron oxidation. It has stalk composed of iron. We talked about it. And the one that I really like is this chemoorganotrophic caulobacter that is famous by not only that stalk is filled with cytoplasm, so stalk in Gallionella is made of iron. Here it's filled of cytoplasm. And interesting about stalk bacteria is they actually have very unusual asymmetric cell division. So they have usually bacteria divide by binary fission and you get two same cells. This one actually divides into two different cells. And the way how it goes, like, so there is one swarmer and one stalk cell. So stalk cells literally divide by elongation, and elongation, like it grows, it's followed by binary fission. So it divides in two. After binary fission is completed, opposite of stalk, you get flagellum to form, and that's now swarmer cell. So that flagellum helps cells to swim around. Cell finds the surface, attaches, stalk forms, flagellum is lost. So usually you have stalk cell and the swarmer cell. Stalk cell is the one that attaches, swarmer cell is the one that has flagellum. So, and this is basically, apparently stalk formation is required for cell division. And there is tons of proteins which people who are in my 311 class will now learn about because that's in their exam question. They will learn about those stalk proteins. So, and we'll see that now. You guys won't, but somebody what I have very many papers on this topic. And the last but not least are bioluminescent bacteria, very interesting organisms that are in the genera Vibrio, Allovibrio, and Photobacterium. Most of them live in marine environment and they are part of marine organisms. They colonize those so-called light organs of marine organisms, and they produce light for the animals, so animals use that light for a signaling. And the way how they produce this light is thanks to the enzyme luciferase. How many of you heard about luciferase? That's what we use in screenings in molecular biology. So this is where it came from these luminescent bacteria. So luciferase oxidized aldehyde reduces oxygen, but most importantly produce light here. So that's what we exploit now. So many luminescent bacteria. So this luminescence actually happens as a response to the high population density, because bacteria don't produce luciferase all the time, but when there is like a high concentration of these inducers, AHL, then AHL cross the membrane and turn on genes for luciferase. So that's like a quorum sensing, if you heard about it, but we don't have to know that here. And this is where I will stop because there is a summary that I want. Yes. Yes. They keep on growing. They keep on growing, yeah. But it always has to be first stalk formation, then binary fission, then swarmer cell, attachment, stalk formation, division. Yeah, thanks. We will finish with summary next class.